Thank you. It's an honor to speak here, although it is a tough act to follow after Darby's talk. I'm going to try to talk a bit about our work in translation of cancer genome with emphasis on in the progress in the area glioblastoma as requested by the chair. What I'm going to talk about is I'll start with some background in genomics, and then I would use the data from the GBM project in the TCGA, the Cancer Genome Atlas project, as a background to try to highlight a few lessons we have learned rather than telling a single story. And I hope to share some of the insights and lessons that we learned over the last decade in how genomic data can be translated into applicable endpoints in the clinic, such as insight that will lead to diagnostic and therapeutics. I think we believe that cancer now, there's sufficient data to make the statement that cancer fundamentally is disease of the genome, or the epigenome. I sort of group all that changes in one word. And therefore, the belief is that if we could understand everything that's going wrong in a cancer genome, we can do a better job at treating the patient that have the disease and prognosticate their future behavior of the disease. More importantly, perhaps do a better job at detecting them early, because there's no question the early detection has the biggest impact on survival. And of course, at the end of the day, what we really want to do is prevent it altogether. Now, the proof of concept that I like to use as an example is actually a story published in 2002 by the Wellcome Trust Sanger Institute Cancer Genome Project under the leadership of Dr. Mike Stratton, Andy Futuor, and Dr. Futuor have fortunately joined us from UK and now a faculty at MD Anderson. So we are really happy to have him be here with us. The Cancer Genome Project team at the Sanger has been pioneers in showing how systematic cancer sequencing can lead to insight that have tremendous impact in management of patients. What they have shown in this study is through sequencing, identify high-frequency mutation in this gene called BRAF, particularly in melanoma patients, and 8 years later, that led to the development of a BRAF inhibitor that ultimately received FDA approval last year and represented the first drug that showed dramatic efficacy. Greater than 80% of the patients with this mutation will respond. And this is the first efficacious drug for advanced melanoma ever. So I think this is a tremendous proof of concept that if we could identify the right targets in the genome that's altered and that the tumor requires, have the right drugs that can inhibit that target, and have a way of selecting the responsive or susceptible population. In other words, the patient that have that mutation, but not the patient that have a different type of mutation, such as NRAS mutated melanoma. Then, putting these three together, we'll be able to deliver a personalized, effective therapy against that disease. So this is really the paradigm of personalized cancer medicine. So how can we take this example and accelerate its implementation in diverse tumor types? So obviously, the first thing we have to do is try to enumerate all the potential targets that we believe alter in the cancer genome that can be the starting point of developing personalized cancer medicine. And that really is the motivation behind the International Cancer Genome Consortia, which is a project involving more than 40 different there are more than 40 different project teams now from over 15 jurisdictions with the goal of sequencing and fully characterizing 20,000 tumor genome in the 5 years period. And that includes both the tumor genome, that's 20,000 patients, that includes the tumor genome as well as the inherited germline genomes. The ICGC the US project, the Cancer Genome Atlas, TCGA belongs under the umbrella of ICGC, and together with other countries and other projects, agreed to a coordinated design and data quality control and so on, so that the data that are generated across the globe can be compared, so that we can begin to understand, for example, how genetics or genomics of breast cancer may be different in different geographic regions or different ethnic groups. So this is an ongoing project. Now, what makes this possible? You just heard from the previous talk that sequencing one gene, or trying to look for one gene, took 16 years? And it's very challenging to do that work, and here we're proposing that they will sequence essentially over 40,000 genomes, 2 from each patient, in a matter of 5 years. And what makes this possible is really the disruptive technology that we call next generation sequencing, or really massively parallel sequencing technology. Just to give you a little comparison data, the human genome was sequenced over 10 years period of time using a Sanger-based method, and that cost a significant amount of money, over $3 billion to do that piece of work. Now, that technology compared to today's technology, the so-called massively parallel sequencing technology, has the kind of throughput that I try to highlight here. The machine that was used, the technology that was used to sequence the human genome can produce about 5 million base pairs of reads per week per instrument. And this is not the latest version of the Illumina high-seq sequencer, can produce over 250 billion bases per week per instrument. In other words, in fact, we know that the single machine output from Illumina high-seq in 1 week can exceed, and did exceed, the entire annual output of all the medical sequencing in 2007 that was funded by NAGI. That is a tremendous increase in throughput, and that explained why the cost of per base pair sequencing have gone down below faster than the Moore's law. Now, with this, it allows us to begin to think about sequencing in a completely different way, given this kind of throughput. More importantly, the kind of data we're getting from massively parallel sequencing technology is much more informative and much more comprehensive. I borrowed this slide from a colleague from this review that summarized how we can think, how I can sort of explain what massively parallel sequencing technology is. Basically, the DNA is fragmented into small pieces, defined length, let's say 400 base pair, and the sequence will read the 2 ends, and those 2 ends will be mapped back to the germline, or the normal sequence. What you can then identify is when there's a base pair that mismatch to the reference, you identify mutation, and the number of reads you can identify the same mutation at that location give you confidence that you are seeing a real mutation. You can also see small insertion and deletion. And when you have complete loss of the gene, a deletion or loss of one copy of that gene, because this is the digital nature in its output, you can actually distinguish between hemizygous and homozygous deletion. Of course, you can see increased copy number amplification. And more importantly, for the first time, when we can systematically, in a high throughput manner, identify structural rearrangement. What that means is that when the 2 ends of that piece of DNA you sequence, when you map it back to the reference, they don't belong in the physical location you expected them. They should be about 400 base pairs apart. Instead, they're in a different area, or maybe even different chromosome, and that informs you that those pieces of DNA have been rearranged. And that's a structural alteration that the previous sequencing technology is not able to identify. And another area that the next-gen sequencing technology is really enhancing is our ability to identify foreign DNA in our genome, and begin to really understand the role of infectious disease such as virus in cancer. And we certainly know that virus infection plays a role in causing cancer. So with all this data, and there's tremendous amount of data gets generated when we do these sequencing, we need to make sense of them first. And I borrowed this slide from a colleague, try to just on high level summarize some of the analysis we have to do. So first, as I mentioned, we always have to characterize the tumor from each patient and compare it to their normal DNA, typically from their blood, or we call germline, a non-tumor DNA, and that allow us to identify alterations that are acquired, rather than alterations that were inherited, such as the Rett syndrome mutation we just heard about. Although more and more we begin to appreciate that we need to begin to incorporate that variation in the germline to really understand the full biological consequence of any one alterations. But if we were focusing primarily on somatic acquired changes, we can compare the tumor DNA alteration to the germline to identify those alterations. And new analytic algorithms are constantly being evolved to do a better job at defining these alterations. But we know now that every cancer genome contains thousands of alterations, from mutation to structure alteration, rearrangement, and so on. And we believe that most of those alterations are what we call passenger. That doesn't mean they have no relevance, and I'll come back and give you an example of that, but they are not necessarily the event that are driving the tumor development or conferring a hallmark feature of the tumor. So what we need to find is which one of these alterations are what we call driver mutations. In other words, they confer biological advantage that allow the tumor to develop. And the first step that we do is to use statistics, looking for alterations that are consistently detected in a large cohort of patients, therefore representing, reflecting a level of positive selection. And these are analytic algorithms that need to be analyzed. So with this as a background, I'm going to use the GBM projects in TCGA as an example to sort of highlight the challenge and the lessons in translating these type of information that we can generate at high speed today, but translating to something that's practically useful. Now, TCGA, the Cancer Genome Atlas project, is funded by the NCI and NHGRI. It was started in 2006 as a pilot, and then is currently in its phase 2. And it has the goal of characterizing 25 major cancer types comprehensively. And the first tumor that is started in the pilot was actually glioblastoma. And then it also defined the study design where it aimed to characterize these tumors on multiple levels, not just mutation, but also copy number alteration, but also expression alteration or DNA methylation, and integrating that with clinical information such as the diagnosis, treatment of that patients, and survival information, and so on. And this is an enormous project involving many centers, and it's really a large network of coordinated activity. So the glioblastoma data currently is summarized on the far left column, and it compares to the study we first published in 2008, which was the first marker paper from the TCGA. As you can see, we have generated significant amount of information, and that was possible because of the next-gen sequencing, or the massively parallel sequencing technology. Back in the day of 2006, when the project was started, we can only, for example, sequence 600 genes in a cohort of 100 tumor, because we are limited by cost and capacity to sequence that real state. However, with next-gen sequencing, we can now sequence every gene, the whole exome, for example, in over 200 and 300 samples, and we can do that at the same cost or less. And that generates the kind of data we now have. And we also can sequence the transcriptome and do whole genome to identify rearrangement, and I'll give you some snapshot of these data. So first, by looking beyond beyond under the lamppost, which would be the 600 genes that were first defined as likely to be important to cancer, we look at the whole genome. You can see that we begin to identify genes that are significantly mutated that have not been previously implicated in cancer, in addition to obviously the signature mutation that we are quite familiar with. For example, I would just name one, the LZTR1 is a little-known transcription factor that's been implicated in the developmental disorder, DiGeorge syndrome. We don't know how and what it does and what role it plays in GBM, but this data now will motivate that study to understand whether there is a functional role and whether there is a connection between these diseases. Another type of data we're getting from these mutation lists is finding, for example, I can't really point to it, BRAF mutations in glioblastoma. I mentioned from the beginning, BRAF mutation is a mutation that's very common in melanoma, which now has an effective drug against. Mutation of BRAF certainly has not been described in glioblastoma, but clearly we see them in a small subset of patients. It led you to ask the question, would that drug be useful in those few patients that harbor that mutation? So these are the kind of questions that the data will lead us to. Now, another interesting observation is also because we're looking at all the genes that are encoded, we can ask questions like, are there classes of genes that affect a particular biological pathway that are collectively altered? And this is one example, the chromatin modification gene. These are genes involved in altering the chromatin structure that control transcriptions and controls it is part of the epigenomics structure, and it can affect transcription of the genes within that locus. So as you can see, when we look at all the GBM cases and look at the patterns of mutation in all of the 160-some genes involving chromatin modification, we were surprised to find that over 40%, in fact 46% of the GBM cases harbor at least one non-synonymous mutation in one of these genes. And often, as you sort of glance at it, you can see that it appears to have these patterns of mutual exclusivity, and it does trend toward that. And so that it's a pattern that suggests that somehow dysregulation of chromatin modification is biologically relevant to this disease. And now we can begin to dive into and asking question how they might affect the pathogenesis of this disease. Another type of data we generate is what we call copy number alteration, which is amplification and deletion in the genome. And this is just a plotter showing on top in blue the regions, you see that deep valley, the region that show deletion, or on the bottom, the red lines showing region that show amplification. This is a mechanism to change the gene dosage, and we heard about from the previous talk how important gene dosage can be. And so all these regions are presumed to harbor a target that the genome is trying to alter the dosage, either by increasing, in the case of oncogene, or by decreasing in the case of period tumor suppressor, and I'll come back to this. We can also look at the gene expression differences, and the community have looked at the transcriptome of glioblastoma and shown that through hierarchical clustering can define different molecular subtype, and primarily the proneural, neural, classical, and mesenchymal subtype. And these tumors had different histopathologic characteristics, and we can also layer on top of that the genomic alteration, gene mutation and DNA amplification and deletion, to ask whether we see enrichment, and sometimes you do see interesting observation. For example, you can see that within the proneural subtype, which can be divided into two further subtypes, I'll come back to that, you see that there's one group of the proneural tumor that seem to have co-occurrence of IDH1 and ATRX mutation. And these are two genes that are involved in both chromatin organization and the IDH1 mutation involving in citric acid enzyme metabolism. So it led you to ask the question how these may be interplaying in this particular subtype of tumor. And more importantly, because we're generating multi-dimensional data on the same tumor, these same tumor also have promoter DNA methylations profile on them. And when we layer in the patterns of DNA promoter methylation, we can define a subset that's called a hypermethylator subtype, or we call it genomic CIMP, or G-CIMP for short. And you can see that these mutations tend to only co-occur in the G-CIMP positive subtype within the proneural subtype. And interestingly, if you look down here, looking for EGFR mutation, which is a signature gene, receptor tyrosine kinase in glioblastoma, you don't see these EGFR mutation in that subtype. Again, suggesting a relationship that might define the behavior of these. Why would you care? Well, that's because we know that these molecular subtypes actually have different clinical outcome. For example, just keep in mind, look at the proneural subtype and the fact that proneural is divided into both G-CIMP positive and G-CIMP negative. Over the last several years, there has been a number of studies that look at these molecular subtypes and correlate to overall outcome of these patients. And they found that these proneural patients are overall, as you look down here, the purple line on the left side, they do better, have a better prognosis. But when we are able to layer on the G-CIMP phenotype, we can see that the survival benefit of that subtype is strictly driven by G-CIMP, not by proneural molecular subtype, as you can see on the Kaplan-Meier curve on the right. And that tells you that genes that are regulated by the G-CIMP hypermethylator group are probably dictating biological behavior of the tumor and conferring better survival in that disease. I think I'm going to have to speed up here. This is another way we can analyze the data, which is looking at pathway. And what we can show here in red with high-frequency mutation, the EGFR, which I already mentioned to you, is a signature mutation. However, we know that over the last decade, there has been numerous attempts to use EGFR inhibitor for GBM patients. And what this shows here is an actual patient who did worse than standard of care when she was treated with an EGFR inhibitor. There have been a number of mechanisms proposed, including redundancy of other receptor tyrosine kinase, such as MET, that can come on to maintain a signaling through this pathway, or downstream PTEN inactivation that can overcome inhibition of the upstream RTK. But another level of complexity is reviewed for the first time by these deep sequencing data. What we are seeing is EGFR mutation is not the simple V3 characteristic mutation. You see many different types of mutation in the EGFR locus, and moreover, these mutations are overlaid on top of copy number amplification, leading to a very heterogeneous pattern, and leads you to wonder, can we assume each one of these alterations have the same biological effect and therefore the same response? The answer is probably not, and this is just another way of showing it. When we look at the transcript that are expressed, you can see, how EGFR is non-canonical how many non-canonical EGFR transcripts are being expressed in the tumor. Again, each one likely have different biological activity, likely to respond differently to the EGFR inhibitor, and that may be another level of complexity that explains why we do not see a significant response to EGFR inhibitor when we take patients that have simply EGFR amplification and mutation. I'm going to actually skip over this part. I want to come back to this one story, which is looking at the copy number alteration. I show you the plot that show that we see across the genome regions of deletion, regions of amplification. Many of these alterations span a region big enough to cover multiple genes. There are evidence that sometimes there are more than one gene that's being targeted by the deletion or amplification, but oftentimes we think of these deletions as, or amplification as really targeting one target, and then you have passenger that got carried along by the alterations. Now, here's an example. I highlighted, and I was going to show you an example of the microRNA MIR34A being a target of this deletion because MIR34A is a tumor suppressor in glioblastoma. But what I want you to pay attention to is that in this region, the little region covered by the red box on chromosome 1, other genes are also deleted whenever you see that deletion of MIR34A, and one of those genes is INO1. And INO1 is an enzyme, and most people think that's a non-essential enzyme or just an enzyme that have many other copies in the body. Why do I care? It's a passenger mutation. But Florian Miller, a very talented postdoc in the DePino lab, thought, well, maybe there's something to this. Maybe if the passenger gene that got deleted, unintended by the genome, not cared by the cancer, but it reduced the overall dosage of that enzyme in the cell. And if now you come in with an inhibitor, the tumor cell that had lower dosage of the enzyme will be more susceptible to the inhibition than the normal cell that have full dosage of that enzyme. In other words, it may be a way of creating a therapeutic window for any drugs that you might use. And that was the concept, and he went on to show, and this is simply telling you where the enolase is sitting in the utilization pathway for glucose. And this is just a summary slide, and the paper is published, so I'm not going into any detail. To summarize that, what he did show is when you use a pan-enolase inhibitor, he can demonstrate that the cell that has lost one deletion of enolase 1, and therefore only got enolase 2 to support that enzymatic activity, they are killed by the inhibitors. On the other hand, these normal cells that have both enolase 1 and enolase 2 have sufficient enzyme remaining even with the inhibitor that they survive, and therefore proving that you can create this therapeutic window. In other words, by leveraging the information in the genome, we can now identify different way of therapeutically targeting cancer in addition to just simply targeting a driver genes. So I'll end here by summarizing that I try to take a couple examples to make the case that we are now in a new era of cancer genomic. We can absolutely generate a complete catalog of somatic alteration and germline alteration variations in cancer and in patients with susceptibility to cancer. We are becoming better and better at analyzing this data. However, the next step, that's only the first step. The next step, we're going to need to understand what these alteration means, and it's only with that understanding and the mechanistic insight we can begin to use that information to develop therapeutic or diagnostic. Moreover, this is a reminder that when we look at cancer, cancer isn't just simply a bunch of cells that are growing rapidly and not stopping. Cancer actually is a pathologic organ, and it have different features, and each one of this, we can link it to underlying genetic alteration that can be driver of the aberrant phenotype, but you can imagine that many of these phenotypes are not something you can easily assess in a cell culture dishes, so that add to the challenge of validating and translating any one of the genomic insight into from a hypothesis to an endpoint that we can utilize. And that's why another aspect that's very important in order for us to efficiently translate the cancer genomic information is to develop better model system and diverse model system. And we heard about great example of what the mouse can do, and then that's certainly an important model that we need to use. So I'll end by simply reiterating the fact that we absolutely need the genomic efforts, otherwise we'd be like filling the blind elephant, the blind man filling the big elephant. However, let's not forget, that's only the first steps. I just want to make an acknowledgement of the TCGA team. It is impossible to list everyone there. I think there are over 300 names. So I would just simply list it here. The disease working group that has been working in glioblastoma, the writing team, and all the tissue sites who contributed the samples, over 17 of them, as well as the center that were generating and analyzing the data. And I try to mention, I would just name Florian, the talented fellow who did the work on collateral lethality of the genome. Thank you.
Thanks very much, Dr. Chin. Are there some questions?
That's a beautiful presentation, but I have a question. The neuropathologist always taught me that if you look at a glioblastoma in one area, you're going to find one histology. If you look at a glioblastoma in a different area, you're going to find a different histology, and that they evolve. And so how do you account for that when you do this? Do you have different pieces? Do you look, at different times?
Okay, Great question, and it's a debate in terms of the study design of the TCGA and the current ICGC. It was designed when the project was conceived several years ago. We did not appreciate this heterogeneity on the genomic level. So the project was designed without what you're suggesting, looking at different part of the tumor. It's simply looking at one piece of the tumor from each patient and do the full characterization. The data that have shown us since then is that exactly as you said, not just in the pathology variation, but underlying genomics are different. If you take one tumor, chop it up in 10 pieces and sequence the 10 pieces separately, you find things that are common and you find many things that are not the same. So there's clearly this intratumoral heterogeneity. I did not put, some of the slides speak to it, but I didn't emphasize that. So right now, we look at the TCGA project as the baseline reference that provide one snapshot. And one of its limitation is it really did not go deep enough and broad enough to look at heterogeneity within a tumor. What it's designed to do is look at heterogeneity across a population. So what we have, this reference provides us a sense of what the intertumoral heterogeneity is. What we now have gone to is the intratumoral heterogeneity. On top of that, more importantly, the evolution piece. Because I think when you say evolution, to me, that's actually the more important piece. How that tumor evolve or respond to a therapy. And that we can only get to when we start doing longitudinal genomics. And that likely will be the emphasis of the next phase of the genomic effort. I don't know what it will be called, but I think right now we don't have that data, and we need that data.
We're a little behind, so just one more quick question, quick answer.
This is a question. I just wonder, do you know of any projects starting on any similar sort of scale for the role of microRNA in cancer?
MicroRNA profiling or what?
Yeah, well, yes, essentially profiling microRNA in cancers.
Yes. So microRNA profiling, both by array and by sequencing, is part of the TCGA project. So every single sample that I talk about and every single sample, the over 10,000 of them in TCGA has microRNA profile, as well as mRNA profile and methylation profiling. So it's a very complete data set. But in terms of scale of project that cover this level, there's an epigenomic project that attempt to really better characterize the epigenome. There's also a network level project to try to do the next step, which is systematically functional validation of some of these events that emerge. So the NIH is attempting to coordinate effort to try to move these data to the next step to make them more useful.